Okay, so in our last little video, we talked about um, the sort of original physical stuff, the geology and the stratigraphy, how we date things, which is quite new, but also the fossils, which are really important to getting people thinking about evolution and finding evidence for that theory. Then we got a bit more scientific and methodical. And we started looking at structures, so, the anatomical structures and some of these chemical things that was linking animals together in plants. And of course, with the advent of DNA and DNA technologies, it's become really quite not easy, much easier to link animals and plants. I mean, you get a much better feel for what has evolved from who or how, and the details are getting much more detailed. That's not the word I want, is it? Anyway, you know what I mean. Whoops, I'm in the way again. Get me out of the way. So, since the middle of last century, many more techniques have become available to people who work in this field. So, paleontologists are those who collect fossils, and anthropologists love studying human lineages and societies. And there's a whole range of people who work in this space who are linked to anthropologists and paleontologists who are keen to work out how life evolved and how things are linked. So these new techniques have become really important to them. One of the interesting early features was this idea of homology. So in this case here we've got the forearm of a human, a mammal, the forearm of a cat, another mammal, the forearm, the flipper, of a whale, and the wing of a bird. These are four mammals. So we already know there's a linkage because we understand they're mammals. But here's some interesting evidence. These are colour-coded to give indication of where these bones appear in each of these structures. So it's the same structure. It's being used for different things. It's being used for upright walking, so locomotion as we use it. For four-legged walking, so it'd be a tetrapod like a cat. The whale is actually a cow that went back to the water, so its front limb has become this flipper. And the bat, of course, is taken to the air, and the front limb has become a wing. It's all about locomotion. So we're seeing animals diverging, moving away from a common ancestor, taking a common structure and evolving it to suit a new requirement. And of course you see that in flowers too, in plants, particularly the flower. And the flower is simply an evolved leaf. And we see this huge variation in leaves as well, in flowers and the leaves that they've come from. But again, it gives an indication of a common ancestor somewhere for that group of plants. So homologous features are about common ancestors and the divergence away from them. We also see analogous features, and here's four wings. So we've got a bit of a moth or butterfly. It's probably a moth actually, by the look of it. Possibly a pterodactyl, I don't know. The bird clearly with feathers, and we've got ourselves another bat. Now the structures are completely different. So this is no common ancestor and diverging from it, but this is what we call convergence. These four animals have converged on the same idea, flight. It's a solution to competition, it's a solution to movement, fast movement through a space. And so the structure of flight has been evolved at least 4 times. The eye's evolved, I think, 7 or 8 times. So some of these structures have evolved many times in many different animals, and so we see what's called convergence. So there's no common ancestor we're diverging from, but rather we're converging on the same outcome, the same need within our ecosystem, within our niche. Comparative embryology wasn't really available to many people for a long time because studying embryos was, particularly human embryos, was always very much frowned upon. But when we look at comparative embryology, remembering that all life began in water, this should not be a real shock. So, if you don't look at the bottom 2 rows, I'll put my hand over that, it won't affect you much. But our fish, here's got its tail, we can see some gills. Here's the amphibian, it's the amphibian. There's gills in place, look at our tail structure. Here's our turtle, definite tail, gills. Our pig, tail, gills. Our human, tail, gills. So that first trimester, that first third of the developmental process in utero shows this amazing familiarity. In the middle third, we start to see a bit more variation developing in these water-based animals, so they look different to these land-based animals, and then we of course got that last trimester we look quite different. But clearly there's a common ancestor and some evolution going on here because of this very first stage. Vestigial structures are very interesting. With our appendix is one of those. The appendix caseum we don't reuse very much, but whales still have hip bones. They don't have any limbs in the back, but the hip still was float. The hip bones there floating inside that whale. So this is evidence of them being from somewhere else, and we now believe whales went back to the water. So they're mammals that went back. And we can use phylogenetic trees to sort of demonstrate all this. I'm in the way. Get me out of the way for a minute. Get over there, Gary. So here's ray fin fish, lungfish, salamanders, frogs, turtles, lizards, sharks, snakes, crocodiles, birds, and mammals. And here's our lineage. So here's a common ancestor to all animals. There's a common ancestor here where the ray fin fish have branched off. There's a common ancestor here to all of that, where the lungfish have branched off. Another common ancestor sees the last of the water-based salamanders and frogs. There's a common ancestor here between salamanders and frogs. And then we have land animals. We've got turtles branching off from an ancestor here, lizards branching off to this group here, which becomes birds, crocodiles, snakes, and lizards. We've got two more common ancestors in here, and the mammals branch off in that direction. So the mammals were around when the dinosaurs were, which is something people forget. So these things are used to try and track this, and the data is, there's immense data involved in trying to work that out. And then we got DNA, and we got amino acid sequences. So at first we can study proteins, and we can see that the proteins have a series of amino acids joined together that make that protein. This is made from our DNA, it's instructed from by the DNA. And so we can use pig proteins and other animal proteins in humans. So insulin sufferers insulin needers used to get insulin from their pigs because it worked. It's another mammal with a very similar protein structure. So this suggests a common ancestry. Then we got to the point where we could actually do DNA hybridization. I'm stuck in the way again, aren't I? Get me out of the way. We'll leave me over there. So essentially you can take DNA from a couple of species and cause it to come apart by heating it. It dissociates. Then you add those species together and cool them, and they can reform into new species. Now sometimes they reform with their own template, so you've got these two strands go back together. But other times you get a mixture of the two species. And then depending on how hot you need to make it to get them to dissociate again, to come apart, will tell you how closely related those species are. So it's a really nice little technique. And now, of course, we can do even more. We can be even more accurate, and we actually sequence the series of DNA nucleotides. You can see adenine, thymine, adenine, cytosine, thymine, G, maybe guanine, adenine, etc. And we can do that for the entire species for its genome, and then compare genomes using obviously pretty big computers. But that allows us to actually compare directly the DNA sequences, and all new alleles are simply mutations from a common ancestor. So we can find those ancestries and reassociate things. And so taxonomy is changing the names of many plants and animals because they weren't quite correctly fitted, particularly amongst the plants. But also we're getting a much better idea of who's evolved from whom and how closely related they are. There's two sources of DNA. You can get DNA obviously from your cell. That's a lot of nuclear DNA. Whereas the mitochondria and the chloroplast both have their own pieces of DNA, particularly these small plasmids, which are circular. And these allow us to actually study these things much more quickly. So you only have to sequence a small amount and compare those mitochondrial DNA or those chloroplast DNA. And in animals, mitochondrial DNA is passed from mother to offspring. So it allows us to actually travel back in time and we'll see where things have come from and who's related to what. And the interesting thing is, of course, that DNA is actually quite robust. So DNA can actually last quite a long time when preserved in fossils, particularly those that are preserved in cold, those that are preserved in amber, maybe. Good old Jurassic Park. Whether you can actually make anything from them doesn't matter, but you can certainly sequence them, which allows us to sequence old DNA and see how it fits into the modern DNA setting. And that's the end of the evidence for evolution. So there's heaps of information now that we can use when we're talking about evolution and trying to explain how things fit together. Those missing links, we don't need to find missing links in fossils, we find the missing links in the nucleotide sequences.